So, very good evening, everyone. Thank you so much for joining. It's been absolutely a pleasure and a kind of a honor for me to introduce my friend, Vishal Acharya, who is a senior scientist at Institute of Himalayan Bioresearch Technology in Palampur, in Himachal Pradesh. Vishal obtained his PhD from Dr. H.A. Nagarajaram's group from CDFD Hyderabad in 2011, and subsequently he has had his scientist fellowship at IHBT Palampur, and since 2011 he has been there as a senior scientist. Over the past, I would say, 14 years of his career, 15 years of his career, Vishal has an incredible touch of working at the coalesce of network biology, larger interest in the areas of plant immune system, bioinformatics, and he has also developed a wonderful database of non-synonymous SNPs, for humans, where whether or not a deleteriosity could really be checked from those particular SNPs. And that particular database was called as Hansa. In fact, that was also a kind of a web server. And I remember when Vishal published it, although I didn't know him, we had an incredible discussion on this, when he won the Bird Award, Bioclues Innovation Research and Development Award in 2017. And that was when, I met him for the first time in 2017. And from 2017 to 2019, he was there as a core member for Bioclues, where he served towards his social responsibility for development of Bioclues. As an alumna of Madurai Kamaraj University, Vishal Acharya hones his wonderful skills at the interface of biology, informatics, and as well as a wonderful inspirational journey where he mentors his graduate students. He's absolutely active. The recent publication of his speaks volumes. The recent publication of his is in Cell Journals iScience, where his lab has discovered wonderful network biology traits in rice. Without wasting much of the time, I would request Vishal to give his first 15 minutes presentation that will be followed by a wonderful Q&A session. Thank you so much, Vishal, for obliging to my request today. Please. Yeah.
Okay. Very, very, very thank you, especially Prash, that I have been interact with Prash, that with the Bioclues organization at that time. So I will like to start to share slide. So I shall start. Okay. And very, very nice. So is it visible? What is one?
Yes, Vishal. Yes, please. Yeah.
Yeah. So I'm thinking that okay. So very, very thank you. Thank Prash that for the kind introduction, and your association with you, and all, especially Bioclues organization, and I have learned a lot from that one organization, especially this. So I will just try to summarize that how kind of work that my lab had been working on, and I will try to show the some glimpse of how bioinformatics affected me, that I have been affected to bioinformatics. So title is just about that applications of machine learning and network biology. That will be the key words of this one. So just I will go back to my MSc year that I have done my MSc in biotechnology from Madurai Kamaraj. So these are the glimpse of the
excuse me, Vishal, your screen is visible, but it's not in full screen. and it's
Ah,
not moving to the next slide. Yeah, please. Yeah.
yes, 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 yes. So now it's okay. Okay, now,
No,
oh,
no,
my
Vishal.
camera is not in.
No, Vishal.
Okay.
It's not at full screen. Maybe you can reload, stop presenting, and reload the presentation. Yeah, it's
Okay.
not full screen yet. Yeah.
So, is it there? No?
No, no.
Okay. I will look out in my uh, shop presenting. I will try to look again. Okay.
Yeah, no problem. No, otherwise you can stop. You can stop sharing and restart sharing. Sometimes that helps. That happens with Google. Chrome browser. Yeah.
I should share or start please. Okay. Share this one.
Yeah, you can make it share full screen. Yeah. Yeah, now it's
Is it
visible.
okay?
Yeah, absolutely visible. Yeah, thank you. Please go ahead. Yeah.
Now it's okay. Now it's okay.
Yeah, yes, please. Yeah, please go ahead. Yeah.
Yes, yes. Okay. Oh, no, I got something missing. Oh, sorry for this time. Okay. So, this is about how I got my exposure of bioinformatics during my MSc biotech project that I have worked with Professor H. Krishna Swami. And this was telling about how the database can be carried out. That my first MSc paper that was published in 2009. So I have learned that it's a basic. What is the importance of a bioinformatics? Actually, he was more interested in crystallography, but he was telling that how we can go with the bioinformatics because it was at a time of 2005. So it was a big boom at that time. So I have learned a lot from that Professor S. Krishnaswamy, and especially my two esteemed teachers, that Professor K. Dhramlingam, that one system, and also in Professor K. Veluthambi, that was for plant pathogen interaction. So these two shaped my career for MSc, that how I should think about bioinformatics in case of immune system and this one. But then I slowly and slowly, it was just
Yeah, Vishal, please go ahead. Yeah.
so. This is the location that I have carried out my PhD. That doing uh, this one is about my PhD supervisor, that Nagaraja Ram, that he told me about how we can think about work was on the machine learning on the human mutation. I will try in just subsequent slide. So this one I have been encouraged, given that how I can think, pursue and freedom means I have been independent researcher, but important point was how to guide this one so you can think independently with some guidance from supervisor. So I am very a big thank to my supervisor to how to think freely and to get a good quality work. So that I will share. And this one I have been honored with European Cyber Fellowship, that while working my PhD, so that I was working on the human mutation, so that I have been given that European Science Foundation. So this also I have spent for 2 months. So that's really shaped me that how to think positively, how to collaboration and how to make a good sign. So this one also I am sharing this picture, and especially this one my friend in Strasbourg. So this was a very famous institute or Strasbourg, that Institute of a Genomics Institute. That one my friend, he told me that how to proceed with your thought, and ultimately give me that some satisfaction that how to pursue the like this. And then subsequently that after working my PhD year, I've been honored with that Indian Science Congress Association, that in the field of a new biology. That one presenting for my PhD work, the how my work on the evolution. I will then on this one the how evolution of the immune plant system that one. And then this one that associated with Prash, that I have
కొంచెం
met him for
మాట్లాడుకుని
the
ఉంటారా?
first time, that he has that this organization honored me with a bird award for this one. So I very lucky to met him at that time. And this all for the bioinformatics organization, that we were having bioinformatics infrastructure facility that we used to have. So we have got second position for the best application for overall from the Department of Biotechnology, and again in the year 2018 we got the third position for the this one. So uh, and lastly, I have been just honored with Associate of a North 2021 in the bioinformatics section. So these all are just honor, but honor is not the important. Important is what is the contribution? What is the important contribution? How bioinformatics that can help you associate with your sign? Mean how you can think that about big data, how you can think about work. So this work I was being associated with my PhD work. So that first paper, that one my PhD work, that was about how I have got a plan that for the software name Hansa. Hansa is just a Sanskrit name. So this was a Sanskrit name. Why Sanskrit name? Because we were thinking about the thing global and name local. So at that time in 2010, so we were thinking to give some Indian name for this. So Hunt is just like the it will separate milk from the water. So that is what we are talking about human mutation. So human mutation means we will have a list of disease causing mutation and list of benign mutation. So what in human protein? So we have developed a software for the discrimination of disease from benign. So from disease from benign. So what is the important point here? So what we have got what we have got that one, the software we have developed in that was published in 2011 in human petition. So that one was giving 20% accuracy than known as the best known method. Means it was about 10% higher than the best known method. So what the important satisfy me that some human National Genetic Reference Laboratory in Manchester, UK, that they have analyzed popular tool on the Hansa. So what happened? They have suggested the advice to use this tool as one other tool. So that one gives immense satisfaction that whatever you have developed, it will be give a quality work, so that other group in UK or somewhere will try to analyze this software for the discrimination of a disease and benign mutation on whatever mean for a human genome. So this one was given one important point contribution that how we can think toward the good bioinformatics. So this one was about the software that we have developed, and this is the PHP page. Just for I want to show the picture of this Hansa. That is what I was just making importance of this skill, how to make things local and mean work local and think global for this one. And then I shifted to after that Prashad interview that after my PhD, so I've shifted to Dhaladhar mountain, so that you can see here that from my office space, so we can see this kind of this one. And this one as a CSIR ISBT Kanampur. So I have been joined here in 2011. That my work was mostly work on the mandate. That means on the Himalayan bioresources. So Himalayan bioresources means we have to think about bioresources available at the Himalaya. So I I got what I got uh, what I got here that that we were working on the artificial intelligence analysis for drug discovery from Himalaya. So what we have attempted to discover some drug targets for a particular key target, mean drug for a key target. So we were planning to develop how to integrate that artificial intelligence and system biology. So we have a developed kind of basis on the protein expression data and network data and plant molecular databases. So these kind of work we got very good accuracy of obtaining Himalayan drug molecule, and subsequently it was went to the wet lab for the validation. So this one was very good satisfaction that we are going to we have developed, and that can be went for the experimental validation, so that they can claim that work for the bioinformatics is validated for this one. And another one is so another one was still there was a big limitation, very big limitation that we have developed at that time, that in 2000 means 15 this one work about how uh, drug discovery can be excellent, can we make for the excellent mean we want to reduce false positive whatever we have developed, still there was a false positive hit means we are talking about whatever we are trying to say lead, that means we are talking about compounds. So compound for that particular target should be very much high accurate. But the is it problem is still there are roughly top 10 of the molecule that when we want to examine in biochemical assays, so it will try to give the more and more number of a false positive hit. So this one it was related to virtual screening. So we have attempted to make new software that automated hit identification optimization and optimization tool. That means we have developed that to how to reduce false positive ultimately, that how we can integrate. So this one was about this one was about so how we have designed. It is just I will try to show just general purpose that how we have integrate, that means chemical space mean it is for chemical drugs we want to go for the identify hit, means you are talking about particular particular target, and particular target means you are thinking about all drugs that we want to make a identify hit. So what the innovation here is, innovation here was how to integrate chemical phase with protein phase. So that one we try to integrate with for identify hit, for particular hit you are thinking that how to go for protein space, and then we got that a very big one optimized hit. So this one I'm just to try to tell that how to integrate for the chemical space and protein space. So we have given the name of the software name is AHIVT, and I'm very proud to share to you that for the drug discovery that was published in Just Journal of Chem Informatics, that in 2022, that for case study we have taken for CXCR4, and we found that for identification and optimization it was reached around a very much higher accuracy of 94% and 81.9%. So all other we have also checked on the independent data set. Means independent data set means uh, independent data set mean is it apart from whatever we have tried for a case study, we have tried for other case study like we have considered here androgen receptor, it was also performed well. So this one is about how we thought about from drug discovery from Himalayas. We were thinking to develop that a software to reduce false positive so that it can be continued for the wet lab validation. But still, it is require more and more need for the development. So I think so. This one was the one work about how the project for the AI for drug discovery, and it had been developed from the past 6 years for this, and still it is going on. For so this one is about one work, and this one is recently we went to the paper that network pharmacology. So we have also integrated the network pharmacology with we have considered for the big data with the network pharmacology, and that one was recently published in theoretical biology and medicine. So this one it was telling about what is the importance of DNA damage protein synergistically affect the cancer prognosis and resistance. So so this one was work about how to plan for the network pharmacology, drug discovery. These are related to the how to improve your work with bioinformatics strong background with strong related to this one. So this one is related to how to go with the flow, and this was also directly invitation from the Proteomics Society newsletter India that this work is very uh, good work. So it was a quite honor from Proteomics Society that how to go for the means DNA damage protein. So they have been interested. It will be going to be published soon for this, and. Now I will show next slide is about my passion, that ultimately passion, that one. See, maybe you will see very bad handwriting here. So I guess that maybe somebody know about this one. See what exactly picture is telling. Picture is telling that in Charles we know about the origin of figure, that is Darwin, that was a phylogenetic tree. So what phylogenetic tree is telling, there is only one figure that always try to attract me. See why evolution? What is the importance of evolution? I think that people always try to understand. So we having a big data, we having a big genome. We also think about how to go with your choice of a genome, go with the choice of Himalayan bioresources. So why not I can apply on the Himalayan bioresources to go for the evolution study. So that attract me very first. So this one was about that how I got evolution of immune system in plant. So that one in about in published in 2017. So this one is about just I we what we found alternative immune defense. means whatever that were observed in animal, so in match NCPA that is important for the animal defense system. So what we found that is only one of the plant, that is corn plant. So corn plant. So what we observe that why is it present in green algae and Selenium? So we try to understand the evolutionary history. So like this one, we got a picture that it may be due to horizontal gene transfer of some of Natch NTPA from cyanobacteria, because it was present in cyanobacteria, and to early green plant. But this was all lost in other plants. So it was lost in other plants because we found the reason that they might have extra immune system. That means number of disease resistant gene is more, so it may not require, so it may have loss. So that one evolution is itself is a big picture. So that my work was attractive to Himalayan bioresources, how to go for the evolution and how to go for the attractive package. That's one related to this work. So this was a whole 5 year project for this one. And I will just to share that it is work is going on under web validation. So it will be going to be exciting to see that what exactly that this domain that found in that early green plant, it will show for the function. So we are looking for this one. So it has been long time. So and it is the latest work is how we got this picture for the deep learning algorithm. I side by side, we were trying to work on the project in immune system. So immune system, we were trying to see that what kind of a model data we have. We can have a Arab justice plant data. We can have a right data. So we try to integrate first time deep learning algorithm. So we have developed this algorithm, and we have tried to integrate expression profile and network data to how to see the network behavior of rice in response to 2 3 pathogen. We want to see that how rice respond to one pathogen, like one is Genthomonas and another one is Menglopod. So these are the bacteria and fungus. So we want to see that how right respond to that one. So we have tried to integrate network biology and deep learning. So this one also try to make a good representation. Good representation means we want to be see that what kind of bioinformatics will try to solve the hypothesis, how we can think. So we will try to say that once we have a data, we can think to generate hypothesis how we can solve using bioinformatics. So this one is only pure dry lab. So we were very happy to develop an algorithm so that it can be applied for other plant pathogen interaction. So that one we have tried to develop this. And I will just kindly acknowledge my two PhD students. That one that was she was working Preeti Arya, that she was working on the evolution of a plant immune system, and that one Vinay Randhawa. Presently, he is at the Harvard Medical School, so he has the work on the software for the drug discovery and how he has developed this one. And one more acknowledge to my all PhD students, especially Neeraj, that recently one paper was
Nimmi Bhavya
AHIUT,
Matlakandi.
and Mithal that on this one DNA damage, and all others that Ravi that is working on the plant pathogen interaction. So I will making more and more critical suggestion. I am thinking that my camera is off or something like this, but I'm not visible. Okay. Anyway, I will just like to thanks for this one, for listening, for patience, and especially Prash that to gave me a conversation. So I will just stop here. It was just I have give a glimpse of all those work, and I thought that it might be a big picture or something like that. So I will just stop sharing the slide here.
Yeah, thank you. Thank you so much, Vishal. That was indeed an excellent one. Friends, I'm sure you'll agree that, this was only a gist. Vishal's excellence is well known. So, Vishal, to start with, I'll probably come up with, an upper chronological order of this wonderful publication of yours in, Cell Life Science. I think I could see that this deep learning network is largely in lieu of biotic stress. So what would be the fate of abiotic stress, and is there any kind of a correlation between abiotic and biotic stress with respect to the networks? Yeah.
Yes, yes, yes. So, yes, the question is about this is about the biotics that we have attempted on, and the biggest problem was when we were thinking about the like plant pathogen interaction. So we were attempt to develop a deep learning algorithm. So data was very limited. So we have tried to develop algorithm that the number of a feature is greater than the number of a sample. So we see that how to think in that way that we should think about the developer algorithm. So earlier it was the algorithm, so we have thought like this: how to proceed with the number of a samples with very less is the number of a feature, because number of feature is expression data. So expression data means you are trying to get 1000 gene, 4000 gene. So this is the number of a feature, and you will get the 60. So the condition was set, at least you can have 30 to 40 samples, so you can work with that one. So that is the one point.
Wonderful, wonderful, Vishal. I mean, let's take this, Vishal. Can this knowledge be translated to maybe the single cell data as well? I know that the network of proteins is largely with respect to whether or not certain proteins with certain biotic stress are known to be interacting with each other, and if they are differentially expressed. So could this knowledge be translated at the gene level, at the single cell transcriptome data as well, Vishal?
Yes, yes. So, now here important point here, you have told that we're starting with at a gene level. So now here we were thinking network data is the input data here.
Right.
So
Right.
network data is the input data. So now the question is, sometime in plant complete protein interaction will not be available.
Right.
So complete protein interaction data will not be available. So complete protein interaction data, if it not available, still we can make to translate that to compensate the loss of a protein interaction by using this algorithm. So algorithm was like this name now, you can say random forest of advanced random forest. So we have used that network guided forest, and we have used a fortnet. So this is the kind of algorithm has been developed earlier. So we have tried to incorporate to compensate the loss of a protein interaction. If the protein interaction is complete and the expression data you can put, then you can give a wonderful hypothesis for the validation. Means you can say that the input data for the ranking of genes, whatever you want to say, that we want to go for the top rank gene. By the machine learning, they will give the top rank of gene. So top rank of gene by incorporating expression data and network data complete. So this will be a good picture about this.
Yeah, wonderful.
So
Wonderful.
that's one.
Yeah, wonderful, Vishal. Yeah, and Vishal, looking back into your wonderful career, In principle, I'm sorry for the background noise. Yeah, in principle, you never had any postdoctoral years of experience, but you had, that wonderful, publication record, publication history, in various fields, right from your PhD heydays. So what was going through your mind? Why didn't you probably go for a kind of an official postdoc elsewhere? Like many PhD folks would do. After their PhD. Yeah.
Very, very nice question. I will just to say that I was also offered that postdoc in Switzerland, Lucerne, before joining here as a scientist fellow.
Right.
But scientist fellow. So I was also in between that I should go or not or whatever. So what now my experience, whatever I'm trying to experience my saying, if you have a very, very good PhD experience, very good PhD means you can say that you are very confident that I can think of the higher height, I can think of the independently. It reflects from your paper or reflects from your mindset. Means what you have thought about high quality. Because I have myself my what I have learned from school of thought. I never thought about the quantity. I always think about it. At least you should have a one high quality paper from 2 or 3 years or 3 or 4 years, but at least. So if you are very able to secure good paper from your side, not from boss support. Boss support will come, but you should think from your own side. If you are able to do, then you are comfortably working in Indian system and you can apply for here. If you are not able to solve this and still you want to learn, so you should learn. You must learn from outside, because I have also experience of going to conference. So I spend 1.5 months from here, and I have learned also uh, learn a lot that okay, how to work. But still, I will get to say if you got the opportunity in India and you are a very good getting your independent thought, so you should start from India. If it not, you should think for the abroad, and you try to think, because going abroad and going India is nothing. It's just the experience how to deal with academic pressure, how to deal with academic publication, and you should think from that side, because sometimes Indian will say ki you don't have any paper. Okay, be patient with you,
కొంచెం
be patient with
మాట్లాడకుండా
you.
ఉంటారా?
That's it. So that is what I know.
Yeah, wonderful, Vishal. I think, well said. I cannot agree anymore. That was a wonderful explanation and insight. I mean, moving ahead, Vishal. I mean, working or finishing your MSc from Madurai Kamraj University. Working with eminent people like KD K Dharmalingam, and of course Velu Thambi, and all those wonderful stalwarts. What was your experience, or what was your what was going through your mind? How good was your you know passion or morale during those particular days? Yeah.
Hmm. Uh, that is why, thank you. That is why I started from MSc, because it was a big inspiration at that time. Because when I joined MSc, that in the first year I could not understand the importance of at that time Shanti Sabru Bhatnagar, or that for A. Dharmlingam, even plant physiology paper from A. Velu Thambi. And at that time I could not understand the meaning, but slowly they, at that time, my senior, my guide is that Krishna Swami, ex Krishna Swami. He himself was a very big scientist from a Purdue University. So that they were very humble at that time. They were telling that how to think about big science. They were not telling. They were just telling to go for the basic course, what excite you, and then go into deep. So when I found a word that bioinformatics and immunology, so I was thinking ki there should be mixing of those. But at that time, I never think that immunology and this was very famous book that Ruby. At that time, QB was very famous book. So that Dr. Dharamlingam used to teach us that how you can say about tall life etc. How to TLR or something like the immune system in 2005. It was went outside of the mind. What that is teaching is there. So sometime I'm thinking that post graduation it should be high difficulty. That is what we are lacking here. That MSc itself is a big to make a bigger choice. MSc is also means you can say post graduation that at that time you can decide what you want to do. So I just influence was they know how to push students and how to push their career, and and we don't know, we were just following their path, and then we realized later they have helped actually a lot how to go with that. That's
No, wonderful, wonderful, Vishal. Well said. That's a wonderful, answer to that. So, Vishal, during the pursuit of your career, were there any disagreements that you put it in an agreeable way with any of your colleagues? Vishal?
yeah, question.
Yeah, yeah. I was asking Vishal, whether you agreed to disagree in any way. Can you just give any example, how you agreed to disagree with your colleagues or with your, mentors or with your maybe with your colleagues, you know? Uh, is there any example that you can cite?
Any example for this?
Yeah. What I was asking is, Vishal, can you cite an example where you disagreed in an agreeable way, with your colleagues or with your mentors?
Mm, uh, I will just say I will just thinking. Actually, I remembering the point. Okay, so actually I was thinking about the question that Anuj had mentioned. But anyway, I was thinking. Okay, chatting. Please go. Prash, please. Can you repeat the question?
Yeah, yeah. Vishal, what I was saying is, did you disagree in an agreeable way on any aspects, with your colleagues?
Or the career for the colleagues. Okay, so so related to passion of what I was also discussing with my students and my colleagues, and what I'm saying to say that passion is very important. That whatever you have a passion for your science is bigger than anyone. So what I am thinking that is sometimes students are lacking about motivation. They just want to finish work. They never try to understand the science in a simple manner, because that have to be work with a complex thing. But the complex thing working will give you very simple answer. So have to think that if you are giving time to your sign will give you back. If you are not giving time to sign, so sign will not give you anything. So just I am thinking, just I will have a one only point to peer my friends and myself also, that I always try to understand sign more and more complex manner, so that I will find a simple thing to be very happy, because depression is very problem, big problem there, because people are not thinking about science. People thinking science as a office work. It is not a office work. It is a passion work. If it's a passion, it's more. So you will give a very beautiful insight, and insight will give you calm you down, and you can work whatever you want, irrespective of your boss is there, irrespective of your director is there, whatever is there. But give time to shine, and that is my idea.
Wonderful, wonderful, Vishal. Yeah, excellent. Vishal, I'm just curious about that Hansa web server. So, keeping in view of the adage of the growth of the number of mutations or variants that have come ever since you published that particular paper, because that was a good old paper in 2011. So, is this database or web server reinstantiated from time to time? Is it updated from time to time?
So uh, database is about it should be updated. That is what you are trying
Yeah,
to say.
the Hansa database. Yeah.
Yeah, the Hansa is a software. That is not the software for this one. So you are trying to say that the database should be updated and it should be like virtually online. That is what we have to think in that way.
Okay. Yeah. Yeah. Wonderful.
Yeah. So Hansa database is not Hansa database. Actually, what I'm trying to say, Hansa is a software database we have used. Yes, it has been reinstated from time to time, but here is itself I am working on it. What problem was when my PhD supervisor has shifted to Central University of Hyderabad from Center for DNA fingerprinting. So there was, and I came here, and there was a problem for the shifting of that database to this one. So it was working up to 2017, up to 2018. So in 3 years, I have asked my supervisor, PhD supervisor for this one to work. So I am again making here to restart this one. So for the software because it should not be soft because other software people have to use software, so it will be very careful for citation and it should be bring down. So that is the one missing point in Indian, that when they are shifting outside, okay, no problem, that work is published and I am here, and that had to be taken care of my previous one. So it should be reinstated. That
Right.
is what. So
Right.
it was working up to 2017. Anyway,
Right. Right.
I got your point.
Wonderful, wonderful, Vishal. So, Vishal, moving further with with the audience questions, we have two wonderful questions. One is from Anuj. Anuj asks, "Will the immune improved plants be can they be considered as GM crops?"
Yeah, okay. So, Anuj, the question is about the immune improved plant category as GM crop. So, GM crop is genetically modified crop. Genetically modified, generally, it can be, but it can be considered. But still, in India, GM crop is very tough to categorize. Means it is still under consideration, but uh, practical implication will be that can be incorporated for the modification of a GM crop. That can be, because this is only for the ranking of gene, because people are using just selected gene and they will start with the work. What we are trying to do, make the bioinformatics deep learning with the ranking of genes. We have tried to explain the network behavior. We were not trying to look for the immune plant. I will just to say one interesting observation. What we found here, that in we have a two kind of pathogen. One pathogen was bacteria, that is we have a Xanthomonas, and another was fungal Magnopotho. So Magnopotho is known as very devastating. It's a very big pathogen. So it's a very means drastic effect to rise, drastic as compared to bacteria. So what we found that using ranking of gene, when we try to understand the right behavior, we found more independent module. Mean whatever the module we found independent, that mean when pathogen attack, so one will be finished, another one will compensate. So this will one another one will compensate. So that kind of behavior means, so if in case of a gentleman, we found compact. Compact means it was a very it's one together. So it will can be broken very easily uh, as compared to gentleman. So what we are trying to say, that the right
Bhavya Matladako.
adaptation is very different, vary from pathogen to pathogen. So that was interesting observation. And one more observation was the this orderness. This orderness means we are trying to say about the IDP, means intrinsic disorder protein. So that flexibility of the protein interaction with each other, that is very less in case of this one magnetoportho, because that one is one not able. So this is one we found. So this one we were talking about the behavior. That is what we try to say hypothesis. We are not trying to for a genetically modified, but we want to say how science of rise and how we can make a hypothesis. So that one. So we are working on another direction from from Genthomonas point of view. We will see the fungal also now. We will see the Genthomonas. This
We are
one only
మాట్లాడగా
about right
చెప్పానండి.
behavior. Now we will want to see the this fungal behavior. So we will dual transcriptome study, dual this one. So this one is set.
Wonderful, wonderful, Vishal. Moving further to Indrani's question, Indrani asked, would this be, deep learning approach be applied, for, drug designing in animal models as well? Your approach here.
Yes, yes, yes. Uh, so, uh, so question was it about what deep learning applicable for drug design and animal sciences.
Right.
So, yes, it can be because that is what we are trying to work on the common platform. We only design for the data. We only play with the data. The data can be anyone, plant, human, or animal, whatever. So, question will be how we can say that what data and sample and ultimately important is hypothesis. What hypothesis you are trying to think? So like you are like deep learning applicable for drug design and animal science. Drug design, we this is a different one. Machine learning, and I'm saying that it may be the mix up. So if we design for the machine learning for the drug design, so drug design we have a random forest, and we should think about what exact feature is there, what feature for biological data is giving. Feature for biological data means if you are thinking about drug target, we should think as a chemical space. Chemical means chemical drug, so we can see as a chemical space. So we should look for the feature to understand the chemical space. Then we should think for the protein space. Why protein space? We were thinking for the particular thing. That was the complexity. We were trying to integrate both chemical and protein space. So that can be applied for the other means case study. Means case study can be shifted, but still you can make a modification of if you want to publish in very high journal, you have to try to modify methodology and modify this one. That is what we have designed for this. So it can be.
Wonderful, wonderful,
But
Vishal.
but we should understand the basic principle of machine learning, machine intelligence. That is what we are working on. Machine intelligence means not only machine learning, deep learning integrated, and then we can think. That's it.
Wonderful, wonderful, Vishal. Anuj, you can unmute your microphone and please ask a very quick question, please. Yeah.
Am I audible?
Yes, please. Yeah.
Yes.
Yeah. Thank you so much. First of all, thank you so much for that interactive session and all. And I have actually I have two questions, small questions. That first is that do we expect some kind of immunological responses, somewhat related to that animals have, because we are just treating the plant systems as well. So the first question
Okay.
is this, and the second question is this: that what prompted to Dr. Vishal to go for this in silicon computational biology work? Although there are lot of work in this that field is going on, and mostly all the labs in India are just focusing more on this wet lab, because there is a school of thought that these dry lab or in silico experiments are not that much relatable to the wet lab experimentations. So I just want to know that what initial thoughts do you have to go for this computational biology and structural biology type of work?
Okay. So, question is related to uh, again repeating like this deep learning, machine learning, and you have an animal kind of you are trying to say that animal pathogen you are trying to study for this animal disease like this. So what we will say, that algorithm algorithm is about uh, what kind of biological data we are using. We have used only public available data. We were using that one. So what we have public available data, we should think what is the threshold should be there. What is the threshold data you should consider for your pathogen data? What is the threshold? To mean what we are thinking that in case of just like animal human data, it will be a very big data. Means we can have a don't have any limitation. So that work can be worked on the this data can be worked on this one. So that is, I'm saying that Anuj maybe not satisfied, but still I'm saying that it is repeating that it can be work that on the plant data it is very tough. We have to justify it. But in case of human data, when we are thinking about interaction data, so it is complete data here, complete interaction data or complete human data, but still error is there. But still we can think about a compensatory behavior of all this one. Mean we should think carefully, suit the interaction, carefully suit the data set, carefully suit the algorithm. We should be think of intermixing should be carefully.
Right,
That's
wonderful,
it.
wonderful, Vishal. And another thing is, Anuj, if I may just put a word, great people always look up into the challenges. They don't work into the smooth parts. Just because all Indian scientists are all working in those particular areas. We should always take up challenges in life. Those challenges would always allow us to reach pinnacle of success, and that is what Vishal has really taken up wonderfully. And it is not a kind of an easy task to take up the computational models. You need a validation study, you need to come up with a cross validation, you need to come up with a precision, you need to come up with a good recall, and all those kind of knitting those particular things is never easy. And I think
Yeah.
Vishal has wonderfully proved that. Thank you so much,
Sorry,
Vishal.
sorry for interruption, and that's why I have only asked that how he can get that courage to go for this kind of work. Because
Right,
to
right.
do this kind of work is always challenging. So that's why I am asked that there are some initial hiccups in the minds of Dr. Vishal as well, whether I will go for this type of work or not. Because
Right,
yeah, that's
right,
why.
Yes,
Thank you
yes,
so
yes,
much for
yes.
the insightful talk. Thank you so much.
right. Wonderful.
You can mail me, no problem.
Yeah.
You can mail me. I can
Yeah.
explain also
Yeah.
whatever
Vishal
you
is
feel
always
comfortable.
pleasant. You can always email him, please. Vishal at ihbt.res.in. Okay, he's always there. So friends, I need to stop here, but not before we take a quick photograph with Vishal. So I would like to request everyone to switch on your camera, and I would like to
Ah,
thank Vishal for this generous time and energy that he has put today. Is always a wonderful scientist, a wonderful scientific person, a wonderful researcher where he comes up with extemporaneous thoughts. His research is always spellbound with wonderful ideas. I always cherish discussion discussing science with him. I remember in 2017 I received him in Jaipur railway station in 2017 as a part of Bird Awards, and that was when I was there at Birla Institute. And he was always smiling, always very close to what every scientist think of think out of box. Thank you so much, Vishal. Yeah, a quick photograph, please,
thank you.
everyone. Yeah.
Uh,
Yes.
Actually, I have to rejoin again for the camera is
Oh,
missing
please,
here.
please, Vishal. We'll wait. Yeah, please rejoin. Yeah.
So can I rejoin? No
Of
problem.
course,
Can
of
rejoin.
course.
Okay.
You are our photograph boy today. Yeah. So we'll wait.
Uh, so I will rejoin again. No problem.
Yes, yes. So we'll wait. Yeah. Yeah. So I'm the photographer here. So Yes, all lovely smiling faces. Thank you so much, everyone. Sugu, Vishal, Anchita, switch on your camera, please. Uh, Anuj, yeah. Abhijit, yeah. If you have camera, please. Yeah,
Oh, again camera is not
yeah.
there.
Good.
Okay.
So say cheese. I'll count three, two, one. Okay. Three, two. Oh, Vishal is not visible. Vishal, you are not
Yes,
visible.
yes. I'm thinking. Have to restart. 1 minute. Okay. Sorry.
Yes, yes, this is the photograph that
Yes.
everyone would really look for. But
Yeah.
I thought, we need. I thought you have taken a patent, for your photograph. You're not. Your face was not visible. Thank you, Vishal. Thank you so much. So three, two, one.
Oh,
Thank you. Another one, please. Three, two, one.
thank you.
Thank you.
Okay.
Thank you so much, Vishal. Thank you so
Okay.
much for
Thank
your
you.
time and energy. Yeah. Namaskar. Thank you so much, everyone. Yeah.
Thank you,
Thank
Vishal.
you.
A huge
Thank
round
you.
of
Thank
applause
you.
for all.
Thank you.
A huge round of
Thank you
applause.
all. Thank you.
Yeah. Please.